Hello, and welcome once again to another episode of Interesting Stories in the History of Diagnostics. This is Mickey Urdea for Halteris Associates. Today I'm going to talk about CRISPR. CRISPR has been in the news, there's a lot going on, and I thought it would be very interesting to talk a bit about the history of CRISPR. Where did it come from? What were the observations that led to where we are? How does CRISPR actually work? And where is the field going? Where is it going in therapeutics? And more interesting to me is where is it going in diagnostics? I found it very interesting that I couldn't find a place where the information about CRISPR that I want to share with you today is all in one place. It just doesn't exist. So I hope you find that the way that I put this together is presented as useful and interesting to you. So I'm going to refer to this technology as CRISPR right from the beginning, but I just want to let you know that it wasn't called that initially. It had a number of names, and I'll come to the point where how it got settled on to call it CRISPR. But this started off with observation in Osaka, Japan in 1987, having to do with the accidental cloning from E. coli of a set of sequences that looked rather weird. They had these interspaces with these short palindromic repeats within them, and people had never seen anything like this before. And it was an interesting observation, and they had no idea what the function could be. And we kind of were stuck in that mode for a while. In 1993, a group in the Netherlands also made an observation about these odd sequences that they found in Mycobacterium tuberculosis. And again, they were very surprised to see how many of them there were. And they differed from one strain to another in terms of the composition between these spacers and the spacers. They didn't know what was going on. So again, an odd observation. In 2000, a researcher in Spain named Francisco Mojica was studying archaea. So we were looking at bacteria before that, now archaea. So we've got these two major domains in prokaryotes that seem to have these types of sequences. He saw these same odd sequences, and he wanted to dig deeper into this. So he and one of his students did a literature search looking for evidence of these types of sequences, and they did this for microbial genomes. This is 2000, and we're starting to actually see some of these sequences appear in literature. And then they developed a small software program to analyze these sequences, and they found 20 different organisms where these repeats existed. These clusters were still very confusing to people. They didn't really know what they were, but Mojica actually had an idea. He thought that perhaps these sequences were involved in some sort of RNA interference that acted as a microbial immune system. A very interesting notion that ends up being correct. So there were a number of people that were investigating these strange sequences at this point. Still didn't know what the function was, still confusing, but they at least wanted to settle on a name. And that name became CRISPR, which was coined by both Mojica and a fellow named Ruud Janssen in the Netherlands. And together they came up with clustered, regularly interspaced, short palindromic repeats. And of course, that's where CRISPR is from. And then in 2005, there's a very interesting set of observations that was made by a yogurt scientist, believe it or not, working at a company called Danesco, and his name was Rodolphe Berenguer. And this had to do with culture of yogurt. And he was working on the organism, the bacterium Streptococcus thermophilus, which is one of the starter organisms that's used in making yogurt. He was having trouble with the quality of yogurt being made with these strains. And he made an observation that when these particular organisms were infected with bacterial viruses or bacteriophage, after several generations, they became resistant to them. And it had some notion that perhaps infections with bacteriophage were the problem in the consistent quality of these starters. And here's the key. It ended up that increased resistance appeared to be associated with the presence of these CRISPR sequences in the bacteria. Still don't know how this worked, but that observation was key. Berenguer was actually able to go back and then modify the sequences in the Streptococcus system that he had originally isolated and replace that with other bacteriophage sequences so that he could generate bacteria that were resistant against additional bacteriophage, which worked out very well. He was able to create a series of strains that, in fact, increased the consistency and the quality of the yogurt starter. So that led to better strains, and Danesco was then finally bought by DuPont, so that has become a mainstay in the production of yogurt. In 2007, there were papers that came from Berenguer and also from the Broad Institute that were the first clear evidence that CRISPR was involved in this microbial immune system. So now the picture's starting to come together. Now the interest is in understanding how is it that the infection of a bacterium with a bacteriophage leads to, in some way, creating that resistance going forward. So a series of studies were carried out by a Dutch group and others, starting to look at the immune system process in E. coli. It appears as though what's happening here is that there is, for instance, in the case of a bacterial phage infection of a bacterium, the double-stranded DNA from the bacteriophage is processed in such a way that it leads to this sequence that will be incorporated into the genome of the bacterium itself that has the parts of the bacteriophage sequence with these interspersed short palindromic repeats. That then is in turn transcribed into RNA. In fact, I'll come back to this, but it's a couple of different types of RNA. And that RNA then gets incorporated into the complex of three enzymes, these what are referred to now as Cas enzymes, Cas proteins. And those proteins then, in combination with those RNAs from the CRISPR transcription, now are capable of degrading the double-stranded DNA from future bacteriophage infections. Okay, so that's what we're seeing there. And as time goes on, you can see that you can do this with RNA as well. And so here's this basic picture. But the point is, right now, this is what becomes referred to as a class 1 CRISPR system, and it's pretty darn complicated. Okay, so if you're going to do this on a routine basis and you want to be able to manipulate cells and use this for future gene editing, not just for resistance of bacteria and archaea to both phage and plasmids, but now we want to actually be able to do this routinely in other types of cells. You need to be able to do something a little simpler than this. So what I just described was de facto DNA interference, not RNA interference, which is what Mojica had originally proposed might be the mechanism here. But not long afterwards, there was another CRISPR form discovered in Porogenes furiosus, which I think is a great name for an organism. They found there that it was a form of RNA interference. So now we have both DNA and RNA. They differ in many ways, and I'll come back to this in a little while. But RNA and DNA can be modified in this way. Now people are starting to really think this stuff through and realizing there's a lot more that can be done with CRISPR than has been done so far. Not just a matter of engineering microorganisms to be resistant to their own pathogens, but there are a lot of other things that can be done with it. And what I've described to date has all been a matter of being able to target and cleave particular pieces of DNA or RNA. But as time goes on, technology is coming about and observations are being made where you can replace them as well. That's really key here. Taking out what you don't want and putting in what you do. At this point, there's a lot of work going on. Many different organizations are looking at CRISPR, and they're finding a remarkable variation in mechanisms of CRISPR, and many different types of enzymes, for instance. Enzymes involved in these Cas systems involve everything from single-stranded DNA nucleases, ATP-dependent helicases, nucleotide cyclases, DNA binding proteins, RNA-guided RNA nucleases, et cetera, et cetera. Now, it's not worthwhile for me to go through a lot of this, but just wanted you to realize that's there. And when I come to diagnostics, you'll see how people are cleverly looking at and exploiting some of those other enzymatic activities in order to create diagnostic tests. So, complications, very exciting. People are very interested in being able to use CRISPR more broadly, but now we need to make this simpler. And now enter two key figures in this whole story, Jennifer Doudna and Emmanuel Charpentier. They were both working in CRISPR. Their labs began to collaborate together. The CRISPR system that I described for dealing with bacteriophage and double-stranded DNA was a so-called class 1 system. It had 3 enzymes, it has 4 different genes associated with it, has 2 types of RNA, very complicated. But what Jennifer and Emmanuel were doing now is they were working on a simpler form of CRISPR from the organism Streptococcus pyogenes. And this is what became called a class 2 system. And this is much simpler. In fact, the Cas enzyme is one large molecule now instead of 3 separate ones. And there are still these 2 types of RNA, but it was significantly simpler than what they had had before. So they were looking at a way to actually simplify that even more. So I mentioned earlier that there were 2 types of RNA. One of them is called CRRNA or CRISPR RNA. The second is called transacting CRRNA. And those two independent molecules were necessary in these class 1 and class 2 systems. And that seemed problematic to the two groups, and they decided to try to come up with a way to make it even simpler. In 2012, the Doudna and Charpentier groups published a joint paper in Science. That paper involved a very careful study of this class 2 system with the Cas enzyme, which is Cas9, by the way. That Cas9 then coupled with the RNAs. And what they did was they were actually able to engineer one type of RNA from those two types of RNA, which they referred to as guide RNA. So now we have a really simple system, Cas9, and it is one guide RNA. That name's going to carry forward here, guide RNA. And that worked extraordinarily well. They speculated that this was going to be a particularly useful way to do gene editing, more so than some of the other things that were out there at the time, like zinc fingers. And man, was that prescient. In fact, 8 years later, 2020, they shared the Nobel Prize, and it has become actually the mainstay of CRISPR to this day. There are modifications of it I'm going to go into in a moment here, but it really was the difference maker in terms of a system that could be manipulated for therapeutic applications and others. And then in 2013, shortly after that Science paper, in 2012, there came two papers, one from Fang Zhang's group at the Broad Institute, and the other from George Church's group at Harvard, which indicated they could actually use that CRISPR system to manipulate human cells in culture. So we just bounced from bacteria in archaea all the way to human cells. Remarkable use of that new, very simple CRISPR system. Now, every day that goes by, you see some other use of CRISPR in some other organism. I mean, we have baker's yeast, zebrafish, fruit flies, ants, mosquitoes, nematode worms, mice, monkeys, and humans. Very recently, as a matter of fact, the first therapeutics based on CRISPR were submitted to the FDA. It comes from Vertex Pharmaceuticals and the company CRISPR Therapeutics. In fact, Emmanuel Charpentier was one of the founders of that company. They have submitted on the use of CRISPR for sickle cell disease as well as for beta thalassemia. And that's just the tip of the iceberg. There are many, many organizations working on the use of CRISPR for therapeutics. And there are lots of issues to work through. One of the daunting problems in CRISPR therapeutics is trying to prove that CRISPR isn't also modifying so-called off-target sequences, making modifications to sequences that you did not want to modify and you do not know the consequences of modifying. This is a daunting problem that will be very difficult to solve. Some of my favorite applications have to do with potential modification of pathogens that have already infected a person. In particular, there's a group called Excision Therapeutics that's working on potential method to actually cure HIV infections and also HBV infections, two chronic conditions for which we don't have a cure. So that's very exciting, but they're going to have to deal with this off-target issue. So in addition to the explosion of the use of CRISPR for potential therapeutics, there has been an explosion concomitantly and independently in an attempt to use CRISPR for diagnostics. And a lot of that early work, as you might imagine, involves Cas9. But now we're seeing the expansion to other particular types of Cas as well, other different types of CRISPR systems. So two popular ones are Cas12 and Cas13, those being in so-called class 3 and class 4 CRISPR. Now, why is that interesting? The reason those two particular enzymes are of interest to people right now is because they involve the so-called collateral cleavages, or unusual activities beyond the simple cleaving up of the pathogen RNA or DNA directly. They actually will simultaneously cleave just any old single-strand RNA or DNA that's present. And you don't see this until the pathogen-specific sequence is clipped, but once that's done, it turns on this additional activity. So people are using that as an attempt to increase the sensitivity of the assays. And the way that's being done is that they're using short pieces of RNA or DNA that they introduce that have been engineered to have a fluorophore or other type of label on one side and a quencher on the other. And so when that piece of RNA or DNA is intact with those two ends on it, there's no signal given off. But once that single-strand nuclease activity occurs, it's an endonuclease activity, it clips it, releases the label from away from the quencher, and now you get a signal. That potentially can give you an increase in sensitivity of the CRISPR-based diagnostic system. In addition to these collateral cleavage activities, there are others, for instance, with different Cas systems, which involve the cyclization of ATP into what is called C4. And that molecule initiates other activities which can be used in a diagnostic format as well. So there's a lot more going on with this, but just to give you a sense of where things are now, there are diagnostics which are available on the market today from a number of companies, in particular Sherlock, that comes out of the Broad Institute, Mammoth, that comes out of the Berkeley Group, and Proof Diagnostics, also involved with Broad. Those companies have been working on products. There are a few on the market, and they are typically ones where a amplification method is used prior to use of the Cas system for detection. Why is that? Well, it's because to date, it's been very difficult to use a direct detection using Cas only and get the sort of molecular sensitivity that you would get from something like PCR or isothermal amplification methods. So they've solved that problem by using both preamplification with one of those two technologies, followed by Cas-based CRISPR technology for detection. To give you some sense of that, the typical PCR or isothermal amplification methods being used today in a laboratory-based format, sometimes in point of care as well, would be on the order of 10 to the second to 10 to the third copies per sample, which is very good. Unfortunately, with the direct detection using CRISPR to date, it's usually been on the order of 10 to the 6th to 10 to the 10th copies, so way off of where you would want to be, which is why these have been combined. There's an astonishing amount of activity going on here to try to come up with methods which are faster, cheaper, more accurate. And there's a great desire to use the incredible precision and accuracy of CRISPR in diagnostics, to be able to do things such as look at much larger numbers of sequences, as would be desirable for certain disease states like cystic fibrosis or antimicrobial resistance, such as in gonorrhea, as in UTIs, and a number of other places. In many cases, that's very difficult to do with PCR and isothermal amplification, although those methodologies are coming along too. So even after all the remarkable activity that's gone on in the last few years here as a result of SARS-CoV-2 in the development of new nucleic acid-based technologies, using mostly isothermal amplification of PCR, we're still in need of things which are better. Better in terms of the time to result, better in terms of the cost, better in terms of the sensitivity, with all those other things working as well, and then looking at a larger number of sequences simultaneously. There's a lot of hope that CRISPR will come to the rescue here, perhaps in conjunction with other technologies like the preamplification. But I think we'll see a lot more happen with CRISPR alone in not too distant future, and I look forward to seeing that. A second issue that's been generally observed with CRISPR has been the length of time of the reactions, which can be, in many cases, 60 to 90 minutes. That's very long. There's an incredible amount of activity going on to shorten that for products, and I'm sure that will occur over time. But that, coupled with sensitivity without using preamplification, have been the major issues. What we still need are products that have the combination of high sensitivity, very low number of molecules being detected, fast turnaround time, very simple to use by non-laboratorians, very low cost, and also, as I mentioned before, can look at a larger number of sequences simultaneously for some of these very specific applications that require that, where PCR and isothermal have struggled. So I'm very hopeful that all the work going on with all the enzymatic methodologies and detection technologies that can be brought to bear on CRISPR are going to be in products, and products which have the attributes that we need. I'm very hopeful of that. I hope you found this episode interesting and useful. And this is Mickey Urdea for Halteris Associates. Thanks very much, and we look forward to having you join us once again in another episode of Interesting Stories in the History of Diagnostics. Goodbye.